Hello students, this is Dr. Amarpreet Kour, a lecturer from Jammu and Kashmir Education Department. Today I will take pollen pistil interaction and I will try to complete this topic in this lecture. But before going ahead, I have a request to all of you. Kindly keep your books, biology books by NCERT of class 12th handy and open the page number 32. Here you will see the figure 2.12 from A to E. All these pictures, they are pertinent to pollen pistil interaction. Okay, in some pictures you will see the germination of pollen grains, and somewhere pollen tube is entering in the synergid. So all these pictures are important and once observe them. Okay, now I'm starting. Students, pollination does not guarantee the transfer of right type of pollens on the stigma. However, pollination is happening, but germination is not possible. There are some compatibility factors. Okay, for example, we have a type of flower or plant in which only self pollination is possible, but by accident the pollen grains of some other plant, they uh, they get transferred to the stigma of self-pollinating plant. So pollination happened, but they will not germinate. Okay, the pistil, I have already explained in the previous lecture that it is a structure that is attaching stigma with ovary, and in spite of that it is performing some important functions. That is, it is recognizing the correct pollen, okay, whether the right type of that is compatible or incompatible pollen grain have arisen in the stigma. Okay, only after the recognition of pollen grains by pistil there is acceptance or rejection of the pollen growth. Okay, pollen grain growth, and it is followed by some chemical procedures between the pollen grain and pistil. These procedures are actually the reactions. The pollen grains, they germinate on the stigma of a particular plant, and pollen tube, it grows through the germ pore. I have already explained in the germ pore the resistant material known as sporulinin, it is absent, and then the content of pollen grains, they move into the pollen tube. Pollen tube, it grows through the tissues of stigma, then style, and finally it reaches to ovary. And remember that the growth of pollen tube, it is precisely chemotropic. Chemotropic means movement due to chemicals. Okay. In pollen grains, there are two cells, generative and vegetative. Vegetative only functions for the nourishment, and it acts as a supportive cell. But generative cell, it divides and it forms two male gametes. Okay, and during the growth of pollen grain, um, sorry, pollen tube, after the grain, it forms a tube, and the vegetative cell is responsible for formation of pollen tube, okay, in the stigma. And if pollen grains, they are shed at two-celled conditions, at three-celled stage, pollen tube, it carries two male gametes from the beginning. Okay. Now there are three entry points in embryo sac where this pollen tube, or you can say in ovary, where this pollen tube it enters. One is micropyle, other is clasa, and third one is integuments. If pollen tube, it enters through micropyle. It is known as porogamy. And if it is entering through clasa, it is known as clasogamy. And if it is entering through integuments, it is known as mesogamy. Whether it is porogamy or clasogamy or mesogamy, but pollen tube enters into the embryo sac via micropillar end. Okay, whatever is the route, but finally it reaches to micropyle and enters into the embryo sac. And what is happening over here? The first thing that is happening is degenerating of synergid, that you can see in your book, in your textbook. The picture is very clear. Okay, some recent research, it has suggested that the filiform apparatus that are present in synergid, they actually guide the this pollen tube for the entry into the embryo sac. This is like a suggestion. All the steps that include the deposition of stigma of pollens on stigma and the entry of pollen tube inside the embryo sac is known as pollen pistil interaction. Clear? Now there are different types of germination. One, the natural one, and the other in vitro. Okay, in vitro is that kind of germination that we are performing. You people are performing, researchers are performing inside the laboratories. Now, this topic, pollen pistil interaction, it has been completed. Now, I will take artificial hybridization. This hybridization, it is usually performed by the horticultures. The first question is that why we need the artificial hybridization. The answer is because we want a desired variety of any plant. Okay, like we want rice with maximum number of grains, and we want the grain size of rice big, larger. So all these, for getting all these characteristics in a particular plant, there is a need of artificial hybridization. Okay, this technique, it involves certain steps like selection of suitable parent, like desired plant. For example, we have two plants. One is possessing one different character, and other is possessing some other different character. But we want a breed in which both of these characters are present. So we select male from one character and female plant from another character. Okay, so this is the selection of suitable parents. Now, if the female plant, it is bisexual, so it is important to remove the anthers of female plant, because we have already selected a plant with desired characters, a male plant. Okay, so what we are doing, we are removing the anthers, and this step is known as emasculation. After that is bagging. We are covering the emasculated flowers with a bag, okay, of suitable size, or you can also wrap it with the butter paper to avoid the contamination of stigma with unwanted pollen grains. Then we do the dusting of pollen grains of our selected desired plant to the anthers of this emasculated and bagged plant. Okay. After the dusting, again we rebag it. We cover it again. Then fruits are then allowed to develop. And if the flower is unisexual, then there is no need of bagging and tagging. Okay, because it contains only one gender. Okay, with this we have completed the artificial hybridization. Thank you for listening my lecture. In case you want to listen any previous lecture, so you can visit my website, that is www.amarpreetkour.com.